Hello viewers, welcome to another biology paper 2 representation. So this one is not a leakage but a revision. So let me not forget, if you are new to this channel, subscribe, share this video to your friends. So we have the first question, which is saying figure 1.1 shows the stages in the digestion of a protein. So on this part here we are having a protein being broken down by enzyme A to form peptide. Then the same peptide has been broken down by enzyme B to form the amino acid. So the amino acids, these are just building blocks of the proteins, meaning they make up the proteins. Now before I even identify enzyme A and enzyme B, let's try to read the question. So the question is saying complete the table below by naming enzymes A and B. So we have enzyme A. This enzyme A is active in the stomach. Then also have enzyme B. This enzyme B is active in the duodenum. So now before we name enzyme A, let's try to know something about the stomach. So the stomach produces what we call the gastric juice. This gastric juice contains three things. It contains the pepsin, which is the enzyme. Then apart from the pepsin, it also contains what we call hydrochloric acid. So here I'm just going to use a symbol because of a space. Then apart from this one here, it also contains what we call renin. Renin is also an enzyme. This one here coagulates the milk. So that's the function of the renin. Then the hydrochloric acid. This one changes the pH from alkalinity to acidic, because this hydrochloric acid is an acid, meaning it is going to change the pH for the action of lipase. This lipase is an enzyme which digests proteins into peptide. So meaning the answer for enzyme A is just going to be pepsin. Then let's try to identify enzyme B. So as for enzyme B, it is active in the duodenum. This one here digests peptide into amino acids. So we need to be able to identify that enzyme. So now this one here is just going to be peptidase. That's the enzyme, peptidase. So the next one is saying figure 1.2 shows how the activity of these enzymes varies with pH. So we are talking about the pH here. So on the y-axis here we are having the enzyme activity. In the x-axis we are having the pH. So the pH scale, it is an instrument that measures now the pH. When you talk of pH, this one is just the abbreviation of potential hydration or potential hydrogen. Now, by definition we can say pH, it is the measure of how acidic or alkalinity or neutrality a substance is. So the first question, which is Roman numeral 1, is saying which of the enzymes, A or B, is active in alkaline conditions. So we are going to find that the pH scale ranges from 0 up to 14. Now this one is ranging from 0.5 up to 11. So we are going to say from 0 up to 6.9, somewhere there, you are going to have the pH of acidic. Meaning on this side here, we are going to have a pH of acidic up to 6.9, somewhere here. Then the pH of 7, it is going to be called as the neutral pH. Then the pH of 8 or 7.9 up to 14, we are going to call this one as the alkaline pH. Now here they are asking us to identify the enzyme which is going to be active in alkaline. So we are going to say it is enzyme B. That's the answer. Then as for Roman numeral number 2, it is asking us to state its optimum pH. So the same enzyme, which is enzyme B. So when you talk of the optimum pH, this one is just the pH at which enzymes work at the best. So beyond the optimum pH, the enzyme is going to undergo denaturalization. Now, the optimum pH of this enzyme, which is B, is going to be 9, this one here. So at this point here, that is where now you are going to have the optimum pH. So the optimum pH, it is at this point here. Now, what pH are we having here? It is 9. So here we are going to say the pH of 9. That's the answer. So beyond this 9, on this part here, the enzyme is going to undergo denaturalization. So the activity of enzyme is just going to happen from this point here up to the optimum pH. Let's see the next question. Question 2. This question 2 is very, very important. You need to know it. As biology is concerned, this question you need to know it. So it is saying figure 2.1 shows some of the processes and organs involved in the regulation of blood sugar levels in blood. Okay, before I interpret the diagram here, let me just try to read the questions. So as for question A, it is saying, name organ C which detects changes in blood sugar levels. So now organ C. What is going to help us to identify organ C? So as you can see here, we have the blood sugar. This organ C is producing the hormone, which is the insulin. And this hormone is converting the blood sugar into glycogen, which is liver glycogen. Then in addition, it is also producing the hormone, which is the glucagon, which is converting now the liver glycogen back into blood sugar, which is the glucose. So meaning organ C is just going to be the pancreas. That's the answer. As for question B, what effects do glucagon and insulin have on the amount of sugar in the blood? So as for the glucagon, as you can see here on the diagram, it is converting the glycogen back into what? Blood sugar. Then the insulin is converting the blood sugar into liver glycogen. So those are the effect. So you are going to find that when there is small amount of glucose in the blood, glucagon is going to be released so that it converts glycogen into glucose so that it can be used by the body during respiration. So we are going to say glucagon converts liver glycogen back into blood sugar when there is small amount of blood sugar in the blood. Then as for the insulin, this insulin is secreted whenever you are having excess glucose in the blood. So you are going to say insulin converts blood sugar into liver glycogen when there is excess blood sugar in the blood. That's the answer. Then let's see the next question. So the next question, which is C, is saying processes A and B occur in the kidney. Name Roman numeral number 1, process A, in which sugar leaves the blood. So what is that process? So let's see. So blood sugar is entering the kidney. Then the blood sugar for process B is leaving the kidney, meaning it is going back now into the blood stream. So try to pause this video and try to answer these processes. So now what is process A? This process A is called the ultrafiltration or filtration. Now, why are we saying ultrafiltration? This ultrafiltration takes place in the part of the nephron called glomerulus. So in the glomerulus, you are going to find that small substances like glucose, excess salts, and excess water are going to be filtered out from the blood. Then big molecules such as the proteins, blood cells, and plasma, these are not going to be filtered out. So process A is ultrafiltration. Then as for process B, this process B is called selectively absorption. Why are we saying selectively absorption? Because glucose now is getting back into the bloodstream. So it is going to be selected. During selectively absorption, useful substances such as glucose, they are not going to be lost out in form of urine. It is going to be reabsorbed back because it is useful for the body during respiration. So we can say as for A, it is going to be ultrafiltration. Now as for B, it was saying process B, in which sugar re-enters the blood, meaning this one is selectively absorption. It is going to be absorbed back into the bloodstream because it is useful for the body during respiration. Then as for question D, diabetics lack insulin and are treated by having regular injections of insulin. So when a person is lacking insulin, or when the pancreas is not secreting insulin, you are going to find that they are going to inject you the artificial insulin so that it converts excess glucose into glycogen. So now the question is saying, explain why this hormone cannot be taken as a pill. The reason why you can't take insulin in form of a pill, you are going to find that the injection of the insulin makes the medication to be fast in terms of effectiveness. Or we can say that when taken in form of injection, it is absorbed fast in the bloodstream and the effective is going to be very fast. So there are two reasons here that I've given you. So the first one I'm saying, when taken in form of the injection, the medication is going to be fast in form of the effectiveness. Then the second one, when taken in form of injection, it is going to be absorbed fast in the bloodstream. So those are the reasons. Let's see the next question. So the next question, it is saying, figure 6.1 shows the stomach and associated organs. Question A, name R, S, and T. So as for R, this one here, it is called the liver. Then as for S, this S is called the gallbladder. That's the name. So the gallbladder stores bile temporarily. Then as for T, T is called the duodenum. That's the answer. Let's see the next questions. So as for B, it is saying, name the fluid that is stored in S. So I'm from identifying S as the gallbladder. So the gallbladder stores the bile. So the fluid is called bile fluid. Then question C, it is saying, T, which is the duodenum, receives fluids from several organs. Name the fluids that are described below. The first one, strongly acidic. So now which one is strongly acidic here? So the fluid which is strongly acidic, it is the hydrochloric acid. That's the answer. Then the next one, for the next one, the fluid contains breakdown products of hemoglobin. So this one here we are going to say it's pepsin, or you can say bile salts. So here there are two answers. You can say pepsin or bile salts. Then the last one, the fluid which hydrolyzes the fats. So this one is going to be an enzyme now, which is going to be called as the lipase. So that is the answer. So now let's try to look at the last one. This question is coming from genetics. So ensure that you understand as I'm going to explain this one. So it is saying, albinism is a rare condition in humans in which normal correlation is absent. It is deduced by inheritance of a pair of recessive alleles, which is the small letters for M. Then figure 7.1 shows the inheritance of correlation of a family tree of 8 individuals, J to Q. So now, whenever you see a person having the alleles, small letters, which is A, the pair of alleles, this one is going to identify that the person is having albinism, or a person is albinism. Then, whenever you see a person having capital letter A and small letter a, or the capital letters for M, this one is going to show that the person is normal. So when it comes to capital letter A and small letter a, this one means that a person is a carrier. Now listen here. When a person is a carrier, it doesn't mean that he is affected by that disease or is affected by that disorder. So question A is saying, Using capital letter A to represent the value for normal coloration and small letter a to represent the value for the absence of coloration. State the genotypes of individuals J and K. So we are identifying J and K. Now before we identify J and K, let's try to look at the key here. So this circle here is representing the female. Then the square is representing the male. So meaning K is the male, J is the female. So albinism is just a gene mutation. It is not a sex-linked characteristic. So when it comes to sex-linked characteristic, you are going to find that a male can't be a carrier. But on this part here, a male is going to be a carrier. So we are having J being normal and also K being normal. Now, once they are met, they are going to have a child, which is a female, because we are having a circle here. So it's going to be a female. And this female, it is albinism. So now if I say these, they are normal, then I use capital letters, whereby I say A, even here A. If we close these two here, are we going to have a child who is an albino? And that one should be a female? No. So now what are we going to do here? We are going to make J to be a carrier and also K to be a carrier. So A, which is capital letter A and small letter a. Capital letter A and small letter a. So these are carriers. Carriers, they are normal. A carrier doesn't mean that a person is affected by a certain disease. No. So now let's try to close this. If we are going to have a child with albinism, let me just draw this one here to help you understand what I'm talking about here. So this one is a simple way of handling a genetic diagram, but it is not allowed in an exam for you to write in form of this. So here I'm going to have capital letter A and small letter a. So we can say that this one is going to represent a male. And on this part here we are going to have a female. So capital letter A and small letter a. So if this one here is close to this one here, what are we going to have here? We are going to have AA, which is normal. Then if this one here is close to this one here, what are we going to have? We are going to have capital letter A and small letter a. And when this one here is close to this one here, we are going to have capital letter A and small letter a. And when this one here is close to this one here, we are going to have small letter A and small letter a, meaning this one is going to be an albino. So now since N is an albino, meaning here they are going to have the small letters, which is A and A. Okay. So here we are having A, which are small letters. So here we can identify that J is having capital letter A and small letter a. Then K is having capital letter A and small letter a. So those are the answers. Let's try to look at the next question. So as for B, Roman number 1 is saying, although O's phenotype is not given in figure 7.1, what would you expect it to be? Normal or albino? So let's try to assess this question. For us to identify that O is an albino or is normal, we are going to see the offsprings that we have here. So this one is an albino and this one is normal. And O was close with what? Was close with N. N was an albino. So what is going to be the phenotype for O? Is it going to be albino or is it going to be the normal? So the parent, which is the female parent, is having normal. Then the male parent is having albino. Okay, since they have already identified for us that M, which is the male, this one is albino. So meaning we need to use the small letters, which is A and A. Then when it comes to the female, which is L, so a female can be a carrier. Someone who is a carrier doesn't mean that is affected by a certain disease. So here we can have a carrier one, or we can have a normal one, where we say we use capital letters, which is the dominant ones. Now let's try to look at if we use the dominant ones close with these. What offsprings are we going to obtain here? We are going to find that they are going to be all carriers. We are going to have capital letter A and small letter A. So all the offsprings are going to be carriers. If we close this one here to this one here, what about if we close this one here with this one? What are we going to have? So here we can say this one is going to be close with this one. Then here we are going to have a carrier. Even here with this one, here we are going to have a carrier. Then here we are going to have albino child. Even here we are going to have an albino child. Now, if we say that O is an albino, whereby we take AA, since O is close with this N here, are we going to have a child who is going to be a normal here? No. We are not going to have a child who is going to be the normal one. So meaning O is going to be a normal. Now, is going to be in form of a carrier. To make you understand, if we say O is going to be an albino, if we close N with O, are we going to have a child who is going to be normal here? All the offsprings are going to be carriers. Now remember here, we are having a normal child, meaning O is going to be a normal one. So now this normal is going to be now in form of what? A carrier. So you can say normal or carrier. So that is the answer. Because when you talk of the phenotype, it is an outward appearance. So here you're not supposed to write the genotypes where you say capital letter A and small letter A. This one here represents the genotype. Now as for Roman numeral number 2, it is saying, explain your choice in Roman numeral number 1. Why are we saying that this one is going to be a normal, which is a carrier? Why are we saying that? So what we are going to do here, we are going to say, this one here, let's try to look at the offspring that we have here, P and Q. P is an albino, then Q is normal. So meaning if we say O is an albino, all the offsprings here are going to have a condition which is albinism. Don't forget N here is also an albino. So if we cross them here, all the offsprings here are going to be albinos. So to explain this one here, we are going to use now these offsprings here that they have produced here. So the reason it being here, we are just going to say, because the offsprings produced, which are P and Q, are albino and normal. Now if O was albino and was close with N, all the offsprings would have been albinos. So that one is the reason for us choosing O as the normal or the carrier. So the last question, which is C, it is saying, if N and O have another child, what is the chance that they will have a son with normal coloration? So this one here, it is just going to be 50%. Why are we saying 50%? Why are we saying this? You're going to find that the chances of you having a boy child who is going to be an albino is going to be equal to you having the girl child who is going to be an albino. So it is just 50%. This one here can be illustrated by using a genetic diagram. N is an albino, where we can say we are having AA. Then O is normal, specifically a carrier. So once we close these, this one is going to be close with this one here. We are going to have a normal in form of a carrier. Then here we are going to have an albino child. Here we are going to have a normal in form of a carrier. Then the last one is going to be an albino. So you can just imagine that this one is a son and this one is a son. This one is a daughter and this one is a daughter here. So there are two. So meaning for us to find 50% here, we are going to say how many albino sons do we have here? We have 2. Then the total number of offsprings altogether, they are about 4. Multiplied by 100%. You're going to find that the answer is just going to be 50%. So that was the last one. Thanks for watching.